Hello everyone, good evening. So today I'm talking about what is the termination part of replication. What is the termination part of replication? So I'll focus in this video I'll focus the termination part of replication in prokaryotes, mainly in prokaryotes. Prokaryotic cell. So firstly, in prokaryotes the replication is mainly circular, like that. It is one stand and it is another stand. So in this video I am talking about the circular replication termination part, and where everybody know the termination part is the end part of our replication, because the first part is initiation, second part is elongation, and the third part is termination. So in this video I am talking about the termination of replication in prokaryotes, mainly circular replication in this video. So firstly, in every circular replication, in initiation part, two types of fork are developed. It is fork 1 and it is fork 2. DNA B protein just come, and further mechanism, it's breaking the hydrogen bond and forming two type of fork. And why it is called fork? So I'm explain. It's like that. Sorry, let me draw freshly. It's like that. So it is called fork because it's look like a frog. So from here the fork word is come. So we are called here one fork and it is here another fork. When two types of fork replicated and come to each other and face each other, and here for facing of each other, termination is happened. So here position, when the two fork are face each other, the termination is stopped. But the process is not very easy. In this process require some types of requirement also. One is tar sequence, and next requirement is TAS protein. So I'm talking in briefly. Mainly TAS sequence in circular DNA present in many positions. This is base pair between two strands. Here one TAS sequence present, here another TAS sequence present, here another TAS sequence present, here another TAS sequence present. So like that in anywhere in circular DNA, TAR sequence is present, and TAS is a protein. It looks like mainly like this. It is called TAS protein, and TAR sequence, which is present in circular DNA, it's create a signal which bring about the TAS protein. So next I am explaining briefly and focus the process. It is one strand. It is another strand, and DNA B protein just come here firstly and break the hydrogen bond and form two type of fork. Suppose it is fork 1 and it is fork 2, and near many this is the base pair between two strands, and here to here suppose present TAR sequence, and here to here suppose present TAR sequence, and here to here suppose present TAR sequence, and here to here suppose present TAR sequence. This TAR sequence create a signal which bring about the TAS protein. So TAS protein just come and bind with the TAR sequence like that. Like that. And replication fork come in this direction, and replication fork 1 come this direction, and replication fork 2 come this direction. This TAS protein have two ends. This TAS protein have two ends. It is one side and it is one side. Sorry, not end. It's two side. The TAS protein have two sides. It is one side and it is another side. These sides is called non-permissive end, and this side is called permissive end. So here this side is called permissive end and this side is non-permissive end. Similarly, it is permissive, it is non-permissive, it is permissive, it is non-permissive. Similarly, it is permissive, it is non-permissive. One line, it is very important. So listen very careful. When the replication fork face this permissive end, when the replication fork face this permissive end, then this TAS protein simply remove from here. This TAS protein simply remove from here, and the replication fork go on. But when the replication fork face the non-permissive end, when the replication fork is face non-permissive end, then the replication fork cannot remove this TAS protein. This is because here the replication fork is stopped. Okay, in this way replication termination is stopped. So I'm explain it. So here replication fork 2 is come here and simply face the permissive end, and this task protein remove, and this fork is go on and face another task protein of permissive end, and it is also removed and goes on, and it is face another task protein which is permissive end, so it is also removed and it is go on, and here another fork is come in this direction, but in replication fork 1 phase the non-permissive end which is responsible for a stop of the replication fork, so replication fork is stopped here, replication fork 1 is stopped here, but replication fork 2 go on and face this task protein non-permissive end, so this task protein simply removed and face then replication fork 2 face the replication fork 1, so it is stopped here, and here some nicks are present, and further it is joined by ligase. But main question is how the replication fork stopped in, why the replication fork is stopped in non-permissive end, why the replication fork stopped in non-permissive end. So let me explain it. It is task sequence and it is task protein, and it is also base pair, and near the non-permissive end of task protein 2 to 3 base pair just flip, 2 to 3 base pair just flip, just 2 to 3 base pair just flip, and which stand in task protein bind in one stand, and where the task protein is bind this stand 2 to 3 base pair just flip, but the another stand 2 to 3 base pair is remain same position, and nextly the base pair is same normally. Here the fork 1 is come, and here fork 2 is come. Firstly fork 1 is come, and in initiation part we are see DNA B protein, firstly break the hydrogen bond, then fork 1 come and further it is replicated. When the DNA B protein face the flipping of the base pair, then DNA B protein cannot be overtake this flipping of the base pair, so the DNA B protein stopped here, cannot be overtake because the base pair is flipped. A DNA B protein cannot be break this flipping of the base pair, so DNA B protein is stopped. So further fork is also stopped because the hydrogen bond breaking is stopped, so the fork 1 is stopped, then replication is stopped here, but the replication fork 2 is come and face the permissive end. Here the base pair is not flipping, so it is also here also a B protein is come, and it is just remove this task protein, and it is face the another fork, it is called face fork 1, so fork 2 and fork 1 then face to each other, and the replication is stopped here, and here some nicking are happened, which is further joined by ligase. In this way in prokaryotes circular replication is happened. So I hope it is helpful for you. Thank you everyone. Please hit the like button, and thank you.